All right, welcome back. Let us continue now uh, to part three of I think what's really a very, very interesting aspect of the bacteria. And I'll try to convince you of this as we go through section 3.3. We're going to differentiate between the envelope structure, which was brought to us by uh, Graham with the Graham stain. We would have never have known these sorts of things until we noticed, or as Graham provided for us, this huge difference between gram positive and gram negative cell wall. And it's a subtle thing, but it's actually again very powerful. Individually weak, but collectively very strong. And this is again what we see with these bacteria, that it's the pieces and parts and how they come together. It's not so much that the parts are simplistic, it's the overall preponderance of the chemical nature of these organisms that's really quite jaw dropping. And so we'll continue on. The cell envelope is composed of two or three basic layers that performs a distinct function, but together, collectively, acts as a checkpoint or a protective unit: the cell wall, the cytoplasmic membrane, and the outer membrane in some bacteria. Okay, why did I say in some bacteria? Let's look at that. If we look at the gram positive, gram negative cell envelopes, notice that the gram positive has this thick peptidoglycan, and embedded in that are these lipotecoic acids, and they're sort of chain linked, as you can see. And I kind of view it as sort of why we put rebar in cement. It tends to make it stronger, is one of the functions, I would imagine, and the peptidoglycan repetitive nature of this, all set on top of a phospholipid bilayer. See, a lot of the things that we've discussed are coming to play now, and embedded in the phospholipid bilayer are these proteins. They're positioned either on the inside or the outside, or it goes across. And that's not by accident. A lot of these things have these special functions. Now let us compare the gram negative. Notice what's missing is this huge peptidoglycan that we see here. We don't see it except for this very small sort of sandwiching between these two slices of bread. Now, granted, the gram positive only has one slice of bread, but this jam sandwich, or this strawberry jam sandwich, has two slices of bread. What I mean by that is that we have phospholipid bilayers, two of them on either side of the peptidoglycan. We only have one, but look at the difference. I'd say that's almost 10 times as thick of a peptidoglycan on the gram positives than we see in the gram negatives. And so what we essentially have is a systematic membrane that has pores and various things embedded in them, allows it to take up more things, where in this one it's pretty isolated from its environment. It's sort of behind this brick wall. We have the membrane that's porous to the outside, where on the gram negative side it's got the systematic membrane allowing whatever it wants to by design, whatever is floating, and either goes all the way across the membrane or it's embedded on one side or another for whatever reason. And then you have the inner membrane that seems to have the same sort of the phospholipid bilayer structure. So we have a periplasmic space on either side, uh, but it's really interesting if we look at uh, what is conferred now to the gram negative as a result of this huge schematical, huge design difference between these two, and it's responsible for clinically the differences that we see in these microorganisms, these being a little bit more resilient to salt. Look at the peptidoglycan, where in here, salt, well, osmotic pressure, you would imagine this would become more susceptible as an example. And we're going to go through various examples, but this one I think is really important to think about. So, helps determine the shape of the bacterium is the cell wall, of course, provides a strong structural support to keep the bacterium from bursting or collapsing because of osmotic pressure. You see, this is really important. Certain drugs target cell walls, disrupting its integrity, causing cell wall lysis. A good example would be having an antibiotic that's penicillin based. It basically disrupts the integrity by preventing certain components to be made, and as a result of that, it weakens the cell wall. But and there are certain drugs that target this. It pokes holes into the cell wall. It's another type, and so it seems to be the types of drugs are different that affect the gram negatives as a gram positive, which of course. Um, so anyhow, we'll get back to this, but this is the key part. A compound composed of a repeating framework of long glycan sugars, peptidoglycan, and cross-linked by short peptides, the so sugars and this cross-linking of these short, you can see the peptide cross-links here, and alanine, glutamate, lysine, linked to glycine seems to be a bridge, and then we have alanine, lysine, glutamate, alanine, and linked to the sugars as we go across, and that's the peptidoglycan, and it's a quite effective networking feature of the cell wall. Gram positive is a thick, homogeneous sheet of peptidoglycan, which you've seen, is 20 to 80 nanometers thick, contains teichoic acid and lipoteichoic acid. The function in cell wall maintenance and enlargement, and contributes to the acidic charge of the cell surface. It's got an acidic charge. So if it's an acid, it's primarily proton. So we're looking at positive type of charging going on. So it's acidic because 0 pH means it's 100% hydrogen, which is positively charged. And so, I wonder what that confers. The gram-negative cell wall is a single thin sheet of peptidoglycan, about 1 to 3. You notice it's about 10 times thinner. And then the gram-positive, and the thinness gives the gram-negative cells greater flexibility and sensitivity to lysis. And so there's two fundamental different forms. This is what we at least screen initially for, is we do a gram stain to determine what's the nature of the beast. Which of the two draw? You're either, well, there is a third bin, but it's not as anywhere near as predominant. We'll talk what the third bin is here in a second. But here's the gram stain. First, the real important stain, and I don't know why Graham chose crystal violet, but he chose lots of different stains, but this is the one that stuck. Get it? Stuck. Well, anyhow. Crystal violet. First, crystal violet's added, and because of the thickness of that peptidoglycan region, it intersperses throughout, where we have the two phospholipid bilayers and a little bit, just a small, 10 times thinner on the gram negative. Graham's iodine is interesting. Now, the word mordant, don't let that throw you. If you've ever seen anyone lay brick, they have this pallet sometimes that they hold the cement in, and that's sort of like a Morden's palette. So you get the drift is that it's sort of like the cement that kind of holds the bricks or plugs the holes in between the bricks. And so when Graham's iodine's added, that's sort of like that uh, cement. And this is a stabilizer that causes the dye to form large complexes. So it kind of holds or cements the crystal violet into the peptidoglycan meshwork. And and so when you add the Graham's iodine, it sort of holds that crystal violet in place, where the iodine has essentially no effect because there's just no place to put that cement. But we apply it to both because we don't know. Then we use alcohol, dissolves lipids in the outer membrane and removes the dye from the peptidoglycan. But it removes it from the gram negatives because the mordant, the iodine, is successfully hiding or keeping in the crystal violet. And this is the premise. Now, Graham had no clue at the molecular level what was going on, and it's not until more recent times we've figured it out. But uh, the the idea is the initial primary stain, which is the crystal violet, retains, and it's really dark. So it's a purple. It's a positive. It's a purple, and it absolutely decolorizes the gram negative. So the gram negative outer membrane gets sort of discombobulated, and then all of the crystal violet leaves. The inner membrane's okay. Then we use the saffron dye, which is a counter stain. Now, because the gram negatives were essentially ripped of its crystal violet because there's no place for it to hold it, we come back with a vital stain, saffron, and it's what we call a counter stain. The vital stain means it actually sticks to the bacteria. That's all it means. It's vitally staining. Their presence is demonstrated by this counter stain. So purple, even though you put the pink, a very faint pink color of the saffron dye, the crystal violet is so dark and just overwhelming, you don't see it. So it's of no consequence to the crystal violet stained bacteria. However, the unstained gram negatives, the counter stain sticks very nicely and turns pink. So we have purple and pink, gram positive purple, and then the pink or the reddish is gram negative. And so that is the essence of the gram stain. Now, sometimes there's certain types of microorganisms that will not stain with a gram stain, and those are always sort of the nightmarish stuff. If that condition comes up while I'm working in a lab uh, and making a diagnosis, it's always kind of concerning because now you're dealing with Mycobacterium or Nocardia, and both are pathogens, and we have to run a third or well, the second type of stain called an acid fast stain, and that allows us now to stain a gram positive, but bulk of the cell wall is composed of unique types of lipids, mycolic acid. These are very long fatty acid chains found in the cell walls of these type of bacteria. They contribute to the pathogenicity, having these weird mycolic acid. They're a type of things that the immune system has a really hard time dealing with. Contributes to the pathogenicity because of that feature, and bacteria are highly resistant to certain chemicals and dyes. So the mycolic acid is sort of like a flame retardant, or it's a special chemical characteristic that these acid fast bacteria seem to possess. We can see this under a microscope using the acid fast or Zeal-Nelson staining. Now, the acid fast was identified by Zeal-Nelson, Zeal and Nelson, and we see the acid fast staining with the weirdness of, it looks more like a gram positive type of stain and that's what we are looking for. So the non-typical cell wall of archaea, which are bacteria that I'm not too concerned about because clinically we don't really worry about it, but they're composed primarily of polysaccharides. And then other cell walls are made of just pure protein. So it seems like the bacteria of modern day, or the ones that we study more, it seems to be more of a hybrid between the older and the newer, where we have a more complex structure. It does not have the peptidoglycan, and some lack a cell wall entirely. The mycoplasmas naturally lack the cell wall, and it kind of speaks to the fact that it may be what we consider an intercellular type of pathogen, so it uses your cells. Sterols in the cell membrane stabilize the cell against lysis. Now, sterols are sort of like cholesterol, and it tends to thicken. It thickens and makes the phospholipid bilayers a little bit more rigid. Mycoplasma pneumoniae, which causes walking pneumonia, is one that really causes a huge issue. L forms, some bacteria that naturally have a cell wall but lose it during part of its life cycle. So there's a particular part of its life cycle where it goes intercellular and says, well, it doesn't need to make the cell wall, which is expensive. If we don't need to make it, lose it. And so they don't lose the genetic potential, they just turn it off. And we call these L forms, and the role in persistent infections, and which makes them more resistant to antibiotics because, well, they're hiding inside cells. The L forms are also known as the Sam Canyon L phase variants. Now, you can do a search on Sam Canyon and find out what the L phase variants are and the cell wall deficient. But the bacterial morphology is determined by the cell wall, and the fact that the L form has no cell wall, its morphology is different from the strain of bacteria it was derived. So the typical L form cells are spheres or spheroids, where the L forms are rod-shaped bacteria, and Bacillus subtilis appear round when they're viewed by phase contrast. They lack cell walls. So if you just think of the L form as lacking the cell wall, L for lack, it's not what it supposedly stands for. It's really the what they refer to as L phase. But the L form bacteria, that's one of the things that we can think about. Similar in composition to most membranes, except contain specialized polysaccharides and proteins. The gram-negatives have this lipopolysaccharide. Now this is problematic. The lipopoly sugars, so it's fat sugar chains, signaling molecules and receptors, and it's referred to as an endotoxin. Now, embedded in these areas are specialized membrane channels, or porins, that allow chemicals to get in and out of the cell. They're very specific for what they allow because of shape and charge, but porin proteins are really, really important in terms of bringing things in and the normal function of the cell. A lipid bilayer, which protein is embedded, so the phospholipid bilayer serves for energy reactions, nutrient processing, and synthesis. So the outer membrane of these things allow the efficiency of bringing things in. More things you can bring in means you don't have to manufacture it, i.e., spend a lot of energy to just to remake it. So if you don't have to make it because you can acquire it, so be it, and it's probably more efficient. Regulates transport nutrients and waste. So what comes in, we also have to get rid of things, and so that's another way to dump it overboard. Selectively permeable, a special carrier mechanism. So the protein is genetically defined to have a certain shape and function, and it fits very nicely, floats in that phospholipid bilayer because of its charge. So you can imagine the region that's actually embedded in the membrane would have to be a hydrophobic region. So we have these long chains of carbons, And most likely that's what you're going to find in that particular design of that protein, that run that sits in the inside of the membrane, and the ends of that protein that has the buoy effect will be hydrophilic. So the regions or the sequences associated with that protein when it folds, more of those hydrophilic would fold on the outer ends, and providing that orientation of that protein, functionality of whatever it's doing. It could have a channel in which things move in, and is very, very detailed and very specific. The cell envelope structure, the outer membrane of gram-negative bacteria contributes an extra barrier. Of course it does. Resistance to certain antimicrobial chemicals, hey, it just doesn't transport them in. I mean, tough tools. If it doesn't have a way to transport it, it's not going to happen. More difficult to inhibit or kill than gram-positive bacteria because it's very selective. It's sort of a gated community, I guess you could say, where it doesn't allow very much through it, where in the gram positives, it's just sort of a barrier, but there's no real smartness in terms of what it allows and doesn't allow. And so alcohol-based compounds dissolve lipids in the outer membrane and therefore damage the cell. So alcohol swabs used to cleanse skin before certain medical procedures, well, you can imagine it's very effective with the gram-negatives. Not so much for the gram-positives. Technically, too, you're supposed to leave that alcohol on for a period of time, like 2 to 3 or 10 minutes or some amount of time that no one seems to pay attention to. But anyhow, the treatment of infections caused by gram-negative bacteria requires drugs that can cross that outer membrane. So the cytoplasm, we're going to be talking about here in a minute, is interesting because we're going to identify sort of the way it's configured. And because 3.4 is so short, I'll just go ahead and cover it. The cytoplasm, 70 to 80% water. And so that's why I show it as a bathtub with everything kind of floating in it. That's sort of the bacteria cell right there. And then we have sugars and amino acids and salts and various things just kind of floating around, no specialized structures. So that's what I meant by having a bathtub sort of view to it, where 70 to 80% is water, and then you've got your play toys and everything floating inside. So the bacteria chromosome and plasmids, the hereditary material, is one big rubber band. I guess you could view it as double-stranded DNA in a big round circle. And we refer to this as a nucleoid. Oid just means it has resemblance to something. Well, it has a resemblance to a nucleus. It doesn't have that formalized double barrier, the outer part of the nucleus. We don't have that, and so it's kind of just out there. And so it's a big circular. Now the smaller plasmids or smaller rings of DNA, they refer to as non-essential. Well, it depends. If the organism is in tissues and the subject is taking tetracycline, and you have a plasmid that encodes for resistance to tetracycline, I would say that was pretty essential. So I would just put quotes around the non-essential. Confers protective traits such as drug resistance, toxic characteristics, or enzyme production. So I don't know how non-essential those would be. If it's producing it, then it's going to need it. Ribosomes, just like we see in eukaryotes, except they're a little bit different. Now, I'll go through this detail here a little bit, but it's a site of protein synthesis composed of ribosomal RNA, which is about 60%, and protein 40%. Okay, so we got folded RNA and proteins that are folded in such a way, and I'll show you some diagrams. But we separate these very similar to separating DNA in gel electrophoresis. We have polyacrylamide gels that we can separate proteins in, and we can put standards in there and size. And we have a certain unit we call the Sveberg unit, which we abbreviate with an S. And so the two subunits are 30S and 50S. So if you add them together, they're 70S. So you can see that we use the ribosome sort of as a interesting gauge. We look at in eukaryotes, the ribosome size, or in Spielberg units, is larger. It's 80S. So it's about 10 Spielberg units larger, and we can tell. Now, the mitochondria, which through endosymbiosis we brought into our cells, and one of the ways we can determine that is the fact that our mitochondria have 70S ribosomes, where all the other cells in us, the eukaryote cell, is 80S. So isn't that interesting? And so we brought that in. Why reinvent the wheel if it's already out there, and it does it very efficiently as a mitochondria. So here's the large subunit and small subunit. And he said, oh my gosh, I don't understand the math here. 50, 60, 70, 80S when you add these two up. But when they are combined together, they take on a slightly different migration pattern through a polyacrylamide gel. And so the S, the Seeberg unit, actually is 70. And so it's how it migrates through the gel because of its configuration. It is larger, 70S, and but you can see the configuration. So the ribosome serves as a site that we're going to thread in messenger RNA, if you recall, and then the transfer RNAs, which are the decoder rings, which we'll cover in the next chapter as we go in, to form the newly formed the peptico, or the proteins that are being made, the peptide being formed. The transfer RNA has to fit just right. In other words, the codon anti-codon match, and when it does, it forms a unit. We'll go over that here in a little bit. Inclusion bodies and microcompartments used for food storage, pack gas and vesicles for buoyancy. Yeah, they're scuba divers. Store crystals of iron oxide with magnetic properties. Some do. There are bacteria that know true north. I guess that's important to know. You can look at your wristwatch and say, "Gee, this is true north." So these bacteria need to know that for whatever reason. Outer shells made of protein. The microcomponents arranged geometrically, so they're kind of in the most basic unit of what buckyballs, or a gentleman that studied basic forms of life that sort of take on certain shapes. Buckminster Fuller came up with these characteristics of geometries, packed full of enzymes designed to work together in biochemical pathways. So they're sort of like ready to go, sort of like toothpaste. You just squirt it out, and it's all set for that particular function, and we are on our way. The cytoskeleton produce long polymers of proteins. They're usually helical ribbons around the cell, and they contribute to the cell. It's sort of like those peanuts that you have in packing material, kind of keeps everything from getting squished when you go to ship it. Nobody wants to receive a package that's got all the contents like jumbled and broken. So we add the peanuts in there to keep it. Well, it's kind of like that. Have been identified in archaea as well, the cytoskeleton. Well, that wouldn't be earth-shattering to think of that. Unique to non-eukaryotic cells, may be potential target for antibiotic development. Well, they've known about that for a long time. So you're going to do something to block it, so you make your bacteria squishy. I don't know how effective that would be, but endospores, really, really important. The dormant body. So when the cell senses, well, things aren't so good in the environment, sort of like, well, there's a COVID virus out there, we need to wear masks and stuff, and it's not looking too good, so let's go ahead and just kind of form these little spores, and we'll just wait this thing out. And that's kind of Bacillus, Clostridia, and Spores Sarcinia are those that can do that. So the vegetative cell is the active cell, but once it gets an SOS command or save our ship or whatever you want to call it, things might be bad. Then it triggers the sporulation routine where it forms these spores, and they're resistant to drying, heating, and they're hunkering down to survive. So you can see that the vegetative cell gets a signal. It starts to provision forming. So these are going into sort of a mode that is going to form now a specialized area to protect the DNA, and sort of like prepping for the worst here, and starts to form this little endospore region where the other part of the vegetative cell that's not being transformed into a spore. And you can see over time it builds and configures it genetically. It's only triggered when it gets a signal to do this, and then forms a spore. If something in the outer environment now, let's say the COVID virus is gone, everything's cool again, and the sun shines out and the birds are chirping, there it is, and it evolves. And this is sort of what the revenge of the Egyptian mummies have is spores and those nasty bandages, and the people, the folks that aren't aware of that go out there and disturb the bandages. It creates an aerosol. They're breathing in. Next thing you know, they're dead. Well, that's not anything other than the fact that the spores are germinating. And Clostridium, well, let's look at some of the ones that can do this. Anthrax. You can acquire anthrax through spores. That's nice. Clostridium tetani. Everyone, I would hope, has had their tetanus shot, and you want to prevent yourself from getting tetanus. Stepping on a rusty nail in the soil, it's a soil microorganism and may contain the spores, and they'll germinate very quickly. Clostridium perfringens, same sort of thing. Gas gangrene. Oh boy, you want to talk about an odor? That is one for the books right there. You will never mistake Clostridium perfringens. It is perhaps the worst odor I have ever smelled. Clostridium botulinum, or Botox, you can take that to, I do that to make my lips more pronounced. I know it's a fashion statement I make. Of course, I'm joking. But there are a lot that folks say that's Clostridium botulinum is what they actually use for that. And I always thought that having botulism was a bad thing, but I guess I've been shown that's not always the case. Clostridium difficile, or C. diff, Code Brown instigator. It's a bad smell, yeah, granted, and it's explosive diarrhea. I like to say the serious gastrointestinal disease has a much nicer kind of connotation, but let me tell you, explosive diarrhea, spray a wall and you'll be cleaning it for, oh my, it's just nasty. Gas gangrene wins in terms of the smell. This is close. It comes in as a second, and it still deserves the ribbons. Yeah. And so that we're going to look at the major comparisons now of the three that we've been talking about. Archaea, which I'm not too worried that you know about. It's just comparative. We have similar things it's more similar to prokaryotes than it is to eukaryotes. So if you know that, I'm happy. So that's a good thing. They have unique sequences in its ribosomal RNA, which that's nice. Inhibit novel methods of DNA compaction. Well, that's nice. They live extremes, high and lows, and some live on the human body and may be capable of causing human disease. But the last time I checked, we don't have extremely high or low temperatures. A lot of these are. Now, some of them are sort of in high salts, and maybe there's an example, but I really have lost to give you one. So, comparison of the three domains. So I would look at this chart looking at bacteria and eukarya. The 70S size is Feinberg units of ribosomes versus 80, And those are the two I really want you to know, because when it comes down to it, this is how we distinguish between prokaryotes and eukaryotes, is one easy way to do that. Contains unique ribosomal RNA signature sequences. They all do. Eukarya like protein synthesis. So archaea kind of wins that. Uh, it has methylation and various things that kind of protects the DNA. Cell wall made of peptidoglycan. Well, the bacteria win that one. They are clearly the only ones that do that we know of. Cytoplasmic membrane lipids, fatty acids with esters, and we see that in eukaryotes and some different types for the archaea to survive in those temperature differences we talked about. Sterols in steroids, cholesterols and things in the membranes, we usually kind of leave that to the eukaryotes. Nucleus and membrane bound, we see that in the eukarya and in the flagellum. Eukaryote flagellum we see in sort of like ciliary action and cilia and that sort of thing. Sperm cells, that sort of thing. And bacteria, that's their primary form of um, locomotion, and archaeum is something unique to the archaea, and so I'm not going to have you worry about that. So that's what I wanted you to know for part 3. Pretty interesting stuff. You get a chance to dig into it some more, that would be great, just to read and to prepare, but it's uh, more of it's going to be delineated in chapter 4, and that's very exciting. Now I have a video I would like you to watch. I posted it in the first section for chapter 4, and it's the inner life of the cell. If you haven't seen it, you should do so. You'll be changed after you see it. I want you to start thinking more 3D and dynamic than flat 2D and boring. And I'll leave it to that, and enjoy the video when you get to that part. And I'll be posting chapter 4 very shortly.